Upload 2 of these. There shouldn't be a memory issue. I've been pretty good, but who knows? Okay, so if I want, but if we go back up to here, right? I'm hoping this oh, there it went away. Great. So I've got my slope is equal to Km over Vmax, right? So remember back from kindergarten where they taught you how to graph? How do you figure out the slope? Rise over run, right? So you'd have to take y1 minus y2 over x1 minus x2. Do you want to do that? I don't want to do it either. So I'm glad we proved this, okay? So, but now we know that our x-intercept, right, is equal to negative 1 over km. We just proved that using the algebra and the equation. And so we can read that off of here. That looks like that's negative 0.01, negative 0.015, negative 0.0717, something like that. Okay, so 0.017 negative is equal to negative 1 over km. This is good, but it worked out. We have that negative sign because you can't have a negative binding constant. Okay? I mean, I guess it could theoretically exist, but you'd never measure it, right? It'd be something pushing something away. Actually, in my research, we get negative binding constants, sometimes calculated, for things that should be binding. That's always fun. Anyway, so if I take 0.017, I divide it into 1. That's how I would solve for this. I get 59. Looks like we got about 2 significant figures. So my Km is equal to 59. 59 what? What do you think? Your answer is on the board. Micromolar, because that's what was on our x-axis, right? We're taking something off our x-axis, and those are the units on our x-axis, right? They're 1 over micromolar, but we already flipped it. Micromolar. Okay, this is ATP binding. Sure, that's great. ADP. ATP in the cell is about 5 millimolar. ADP, you know, if it got up to about 1 millimolar, you'd really want to use it, right? You'd want to turn it into ATP, and so that's way below. So you're pretty well bound at that point. We'll learn more about that. Okay, questions? All right, so what do? We have our answers up here. We had our Vmax. We figured that out, and that was 50 millimolar per second. Right? And then we did our Km, and that was, I've already forgot, 59 micromolar. All right, next question. Which substrate did we just calculate the Km? I just kind of gave it away. ADP. How would you know it if I hadn't given it away? What? It's in the question. Well, so is creatine phosphate. So I've got creatine phosphate and ADP. How do I know? It's what I'm varying on the x-axis, right? This is how I do this experiment. I vary this. That's what I'm getting data for. Okay? So that's how you know for that. All right, what's our next question? At the molecular level, what does Km tell me about the enzyme?
It's weakly. This is like too high.
Okay, what more generally? What does it tell me? What does it tell me about the enzyme?
It's affinity.
You're telling me it's affinity, right? It's telling me how tightly the enzyme binds ADP,
Yeah.
right? Okay, for this one. My gosh, oh, this is a PDF. This is going to be a mess, maybe. Yeah. How tightly the enzyme binds to ADP in this case. But how tightly whatever one you were varying is what it's going to tell us. What does K-cat tell me about the enzyme?
How fast does it go?
How fast it goes. And it tells me actually K-cat is the turnover number. It's the special number we want to talk about. K-cat, supposed to be a baby K, tells us how fast the enzyme is making product. Okay, but I want to be even more specific than that. All right, we haven't even calculated K-cat yet. Okay, we've got a Vmax. So more specifically, K-cat tells us K-cat is the turnover number. Let's call it that. It tells us how many products a single enzyme molecule will make in a unit given time when fully saturated. And what I mean by that fully saturated is what we talked about earlier. Fully saturated means that every time it makes a product, it immediately gets another substrate instantaneously. Okay. Questions? All right, let's do the next one then. So if the enzyme concentration for these experiments was 5 micromolar, what is the K-cat for the enzyme? Now units matter. Okay? So what we had is we know that our Vmax is equal to our K-cat times our enzyme total concentration, right? So what I'm telling you here is how much enzyme was dumped into the reaction, right? Or what concentration we ended up, not actually how much. And so I can put that down here and I can have 5 micromolar over here, and then I have my Vmax was 50 millimolar per second, and then I can solve for my K-cat. That seems pretty easy. Anything wrong with that? Units, right. I need to have these in the same units. So units must match. Anybody know how many millimolar are in a micromolar? 1,000, right. So I can convert either way to make them match. But what I'm going to do is I'm going to say, so we said micromolar and a millimolar. So we're going to say if I have 50 millimolar per second in 1 millimolar, I have 1,000 micromolar. Okay. Millimolar cancels with millimolar, and I'm going to end up with 50,000 micromolar per second. All right, so then I can come back over here and change this into 50,000. Divide it by 5, right, to get 5 micromolar from both sides. let's make this really clear that I made this into a micromolar. Okay, 50,000 divided by 5, I'm going to end up with 10,000. Kcat is equal to 10,000 per second. My micromolar cancels with micromolar, right? So what that means is when this enzyme is fully saturated, it can make 10,000 products per second. Okay, that's what the Kcat means. That's one could do that. If I had 1 mole of enzyme, it would make 10,000 moles per second. Questions? Yes?
When you plug in the 5 micromolars, is that just, can you plug in any number, or where did we get that from?
We got that from the worksheet. So if we look up here, right here, I told you what that concentration was of the enzyme.
Okay, so you could plug in any number?
You can put any number if that's what it was for the problem, yes. It doesn't have to be 5 micromolar. And you could adjust both of them to whatever concentration you want as long as they match. I think that's the full worksheet, is it? Okay. That is the big stuff that I want you to be able to do. This is the most important. I'm going to put up a bunch of worksheets. This is the most important thing in biochemistry. Like I said before, if somebody asks you a question on one of your big scary tests later or wants you to know anything from biochemistry, and it's only one thing, it's most likely to be this stuff. Okay? What we can go over right here. They probably won't ask you to plot by hand. Okay, questions? All right, then let's go on and we're going to start talking about enzyme inhibitors. Yes? So if you're graphing by hand, I got some answers there. You were graphing on graph paper, so your answers are probably probably better than mine, right? So what I'll do is I'll plot it on. I'll use Excel to plot it to get the answer, and then I'll give you like a 30% leeway probably. So be careful, but I'm not expecting 5% error or anything like that, like I expect on the lab you just did. Okay. Questions? Other questions? That's a good question. So yeah, I'm definitely going to give some slack because you're plotting by hand. All right, I will post some of those. I'll post a bunch of practice problems for that. So the big reason why we're studying enzymes right here is I've talked about this before. Remember we talked about HIV protease and how what happens in the life cycle of HIV. I didn't bring a picture for this, but it kind of looks like this. It's got 2 big proteins it makes. So it actually, on infection, you get these 2 big proteins that are made, and one of them actually goes into the coat of the virus, and so it needs to be chopped up into pieces that can then be used to coat the virus. All right, and the other one makes the enzymes that are important for the virus life cycle, which are a protease, an integrase, and a reverse transcriptase. All right, and so then these things are coat proteins, let's say. And so all of these things are needed for the virus to kind of get going. Okay, so the virus infects a cell, it can't take its DNA genome, or it's got an RNA genome, but it can't take its RNA genome, make it into a DNA genome, and then stick it into your genome, unless, and it has these things to begin with. okay? And then it can't make more of these things unless it has some protease to start with. So these proteins are actually in the virus particle itself, and that makes this a great drug target. And I'm writing it down the board because I kind of feel like it, but also I'm not going to test you on this specific content. This is why enzyme inhibitors are important. This is why we're learning Michaelis-Menten kinetics. All right, we want to know what the turnover numbers are for these enzymes. We want to know how tightly they bind to their substrates, because we want to take something like HIV protease that has to be around at the beginning of the virus life cycle in a cell to cut this thing up so you can make more virus, and we want to make an inhibitor that stops it. Okay, inhibitor that makes it so it can't do that, and the virus can't get started. Or for HIV, if you slow it down, HIV, the problem with HIV infection is that it's the virus is attacking the immune system, and so you have this balance, you have this fight between the immune system and the virus, and over time, eventually the immune system loses, essentially. Okay, to put it in really lay terms there. And so if you can take this and you can take somebody can take some drugs that will slow down the virus, now we've given the immune system an advantage, and then the immune system can actually not win that battle and completely get rid of the virus for this, but it can actually beat back the virus and keep the virus beat back, if that makes sense. That's how these sorts of things work. So this is why this stuff is important. So we're going to talk about enzyme inhibition. So there are two broad categories of enzyme inhibitors. Okay, and those two broad categories are, they're going to be covalent and non-covalent inhibitors. Okay, and so covalent inhibitors, obviously from the name, they're going to bind directly to the enzyme. Okay, and non-covalent inhibitors do not. Okay, they're going to bind reversibly. They don't make covalent bonds. Now, they might bind really tightly, which makes it almost irreversible. And that's a really good drug if you can do that. So, but we're going to focus on these non-covalent inhibitors here. That's generally what happens in biochemistry. And so there are four non-covalent types of non-covalent inhibitors. All right, there's going to be competitive. There is non-competitive. Good guess, or you've seen it before. Oh, you have seen it before. I did not name these, by the way. And then mixed. Okay, so we have these different sorts of things. So these are going to all bind in terms of these three. These have specific mechanisms and specific binding characteristics. And then mixed is kind of can be a mixture of the effects you see over here. Competitive is going to be the most important. All right, so if you leave this class and somebody's asking you on some big scary test, what kind of inhibitor is this, right? And they give you some data and you don't remember any of biochemistry because it's been too long. Say it's competitive, okay? But hopefully you'll remember in case they're being tricky with you. And I'll give you one of the other ones. They're all important. But let's go ahead and talk about these, or we'll start talking about these. So we'll start with competitive inhibitors. Competitive inhibitors are going to bind to the active site of the enzyme. They're going to compete with the substrate for the active site of the enzyme. So they're competing with the enzyme for binding. So if we think about, go back to our Pac-Man model for enzymes. I've got my enzyme here. It has an active site. And there would be an inhibitor that would maybe have this shape, I for inhibitor there. And then we'd have our substrate that would have this shape. And both could bind based on their shapes to the active site. But if the inhibitor is in the active site, the substrate can't bind. So if I was to draw this out like equations, we've got enzyme plus substrate, and that enzyme substrate can go and bind to enzyme substrate, make an enzyme substrate complex, right? And then we can go to make enzyme plus product. But if I have a competitive inhibitor here, I could also have that enzyme bind to this inhibitor, and I would make an enzyme inhibitor complex that then doesn't do anything. So think about this. I've kind of drawn it as, maybe I should have drawn it as straight across like this, okay, rather than down. But think about this in terms of a Le Chatelier's principle. So I've got enzyme, right, and it's got the choice of one of these things. If I add more inhibitor, what happens to my enzyme? Le Chatelier's principle. It's going to make more enzyme inhibitor complex, right? What happens if I add more substrate? I make more enzyme substrate product. Okay, so what this means is we can use Le Chatelier's principle to understand how this works, and you can use it to overcome the inhibition in this case. So if I'm talking about, I've got a graph here where I've got my substrate and I've got my initial rate of reaction, and let's say this is my no inhibitor. My no inhibitor graph. What would happen with my inhibitor graph is if I've got some inhibitor around, it's going to be slower getting to that max rate, but I can still get the max rate if I add enough substrate. So I can, by Le Chatelier's principle, if I keep adding substrate, I can outcompete the inhibitor and get my same max rate. Right? So that's our V max. And what do we remember about Vmax in our equations? Well, Vmax is equal to Kcat times substrate concentration, right? So how do I make my Vmax go up in a reaction for a given enzyme? Oh, I'm sorry, it's not more substrate. That's enzyme total, right? My bad. All right, so I add more enzyme, right? This is a constant. I can't change that for an enzyme, so I can add more enzyme. So what that means, though, is for a given amount of enzyme, if I can just add enough substrate to get my same Vmax, then I haven't changed my Kcat. Okay? So in competitive inhibition, Kcat is unchanged. Do you see why it's unchanged? We can get the same Vmax. No? You don't see why it's unchanged? Okay. So we have, do you agree that Vmax is unchanged? So I can, Vmax is how fast this thing will go, right?
Mhm.
And I can, if I add infinite substrate, which I can't because then stuff would precipitate out, theoretically you could add infinite substrate, right? But if I can add enough substrate, I can overcome that inhibitor, regardless of how good that inhibitor is.
So the second graph is only exists if like there's enough substrate.
You just add more substrate to get to recover your activity.
Okay.
And that's, we know we can do that because of Le Chatelier's principle,
Mm-hmm.
right? These are competing with each other. And these two go together, right? I can raise my Vmax by changing my enzyme substrate concentration, or I can raise my kcat by, if my kcat went up, right, or moved around. But that's for an enzyme that's fixed. And this, in this given graph over here, is fixed as well. So maybe that's a good, important point to make here, is that whenever I draw one of these, this is for a given amount of enzyme. It is not, enzyme cannot vary through that graph. The only things that can vary are, the only thing that can vary is how much substrate you start out with. That make sense? Okay. So kcat's unchanged. Now, how do I calculate my Km from one of these graphs? It's 1/2 V naught, right? So if I follow for my, actually let me make it in green. If I follow for my green over 1/2 my max rate and I follow it down, here's my green Km. And if I then take my gray here and I follow it over and follow it down, here's my gray Km, or my inhibited Km. Is Km changed? Yes. Okay. Apparent, we call it apparent Km has changed. Okay. The actual affinity of the enzyme for the substrate is the same, but because it's competing with a competing with an inhibitor, it looks like the Km it changes. So I'm just gonna say Km changes. Km is increased. Very good. Questions on that? How we get that from that graph? All right, then let's really quick, because I've got your exams to hand back. Let's quick look at how this would look on a Lineweaver-Burke plot. So a Lineweaver-Burke plot looks like this. I have 1 over my substrate concentration versus 1 over my V naught. Let's say, what did we do? We went with green and gray. So there's my green line, right? Did my kcat change with a competitive inhibitor? No, so my Vmax didn't change, right? So my y-intercept shouldn't change. And so it's going to look like this. This is my inhibited, or inhibitor. This is no inhibitor. So if you have a Lineweaver-Burke plot, or you're allowed to get it, what you want to do with that is you want to look to see if the Vmax has changed. If they all have the same y-intercept, then that means that you're looking at a competitive inhibitor. Okay, because that Vmax doesn't change. Why did I draw my gray line above the other line? Because it's the inverse, and so it's inhibited, it's going slower, right? And 1 over slower is a bigger number, because the rate is in the denominator. All right, so we've got 3 more of those we're going to go over in the next lecture. But I think for now, I'm going to go ahead and hand these back. So I have